The Bioterol project was funded by the Marie Curie Intra-European Fellowships for Career Development in the 2013 call. Bioterol is the acronym for biodegradation of tertiary alcohols. In the following slides, I will present the context which leads us to the objectives of the project. Human health relies heavily on access to clean drinking water, but 1 in 10 people worldwide lack access to it. That is, more than the population in the EU do not have access to clean water. So prevention of aquifer contamination is essential. But in the unfortunate event of a contamination spill, we want to control the fate of that chemical. In order to evaluate the fate of a chemical, we must also take into consideration the current and emerging properties of microorganisms, that is, the evolution of microbial degradation pathways. And this will depend on the chemical structure of the contaminant, since it's not the same to degrade a monomer, polymer, branched or unbranched compound. We also have to take into account the evolutionary clock, where time is measured in number of generations, that is, the time it takes for a cell to divide, and how long do we keep this clock ticking. The mutation rate of each individual microorganism is different, and generally higher mutation rates will confer more beneficial mutations, along with more deleterious mutations. Additionally, the selective pressure exerted on the microorganisms will drive mutations towards adaptation of the selective pressure. For example, serial transfers will increase growth rates towards the carbon substrate in the medium. The evolution of degradation pathways from tertiary alcohols will be the focus of our project, where a tertiary alcohol is a compound in which a hydroxyl group is attached to a saturated carbon atom, which has 3 other carbon atoms attached to it. The bacterium Aquincola tertiaricarbonis L108 is a microbial species which was isolated in a long-term fuel ether contamination site in Leuna. It was named after its capacity to degrade tertiary carbon compounds such as MTBE, ETBE, TAME and TAEE, which are fuel oxygenates. When we took a closer look at the genome of strain L108, we observed a supercluster of clusters, all connected to fuel ether degradation. This supercluster is highly homologous to that of another fuel ether degrader, Methylibium petroleiphilum PM1. When we compare each cluster to other clusters in annotated databases, we can tell that most likely they came from different microorganisms throughout different phyla. So this is a result of an evolved species who has acquired pathways from other bacteria and made rearrangements and regulation adjustments. But it arises questions like, how long did it take to get here, and what was its original genetic makeup before this cluster was incorporated and organized? The microbial physiology group at the UFZ in Leipzig have a background in MTBE degradation and tertiary alcohols, and this was our starting point to explore the evolution of a degradation pathway. MTBE and ETBE are hydrolyzed to terbutyl alcohol. TAME and TAEE are also degraded to tert-amyl alcohol. In strain L108, the following enzymes have been elucidated: a cytochrome P450 monoxygenase, a hydroxylase, and cobalamin-dependent mutase. Surprisingly, the enzyme that encodes for the hydroxylase had a double function, as a hydroxylase from TBA and as a desaturase for TAA. So 2 different functions for one sole enzyme. So what was different about these 2 structures? Both are tertiary alcohols. TBA is the simplest tertiary alcohol and TAA is a heavier homologue. And so the TAA metabolic pathway was elucidated. Between 7 to 8 different enzymatic steps were involved in this new pathway. The blue-colored enzymes are well known, and at least 3 new unknown enzymes were awaiting to be discovered, an isomerase and 2 dehydrogenases. So we set out to use these as the example of what could happen if we introduced Aquincola to a new heavier tertiary alcohol. A new tertiary heavier alcohol like 3-methyl-3-pentanol. Strain L108 can grow in TAA with a generation time of 10 hours, and preliminary tests show that increasing heavier homolog compounds to C6 and C7 compounds increased generation times until this was no longer effective. So can we evolve L108 on larger tertiary alcohols and improve current enzymatic rates? And if not, what could be the rate limiting step? And this leads us to the fundamental aims of the Bioterol project, to study the evolution on a tertiary alcohol like the C6, 3M3P, coevolution on a heavier tertiary alcohol, and to elucidate the biochemical pathway for tertiary alcohols. The impact of the project is that by understanding evolutionary pathways, we could eventually model natural attenuation. So we went on a mission to evolve L108 in the lab on the already slow-growing carbon compound 3-methyl-3-pentanol. And there are 3 ways of doing the evolution experiments: single-cell transfers, continuous culture, or serial transfer. And we thought we would first start out with the serial transfers, just like the Lenski lines in E. coli. The idea is that mutations will arise during growth and cells with higher growth will be transferred. This incrementation in growth rate are usually small, so we need many generations to observe a significant change from the parent strain. How many generations? Well, that's one of our questions. Richard Lenski's group set up 12 flasks of E. coli back in 1988. They were allowed to grow overnight in minimal medium containing glucose as the only carbon source and citrate as a chelating agent. Serial cultures were used and the cells grew with approximately 6 generations per day, which meant that after 14 years the cells had undergone 33,000 divisions and had evolved to use citrate as an additional carbon source. When we transpose this concept to Aquincola, the evolution on the C6 compound would take 150 years to obtain the same number of generations that the E. coli took to adapt to citrate uptake. So are we crazy? Well, no. We are not trying to evolve a new pathway, only increase the efficiency of an existing one on heavier homologs. So from our evolution experiment, we will be able to compare changes in the parent strain and the evolved strain at the genomic, physiological, and enzymatic level. In order to determine the evolutionary effects, one must find the genes involved in this pathway, and thus the importance of functional annotation of genes. So elucidation of the enzymatic pathway will be performed using classical gene knockout methods and more innovative techniques, such as proteome profiling. Thank you for staying with us, and please visit the Microbial Physiology Group webpage for updates on the project, which can be found below.